Hello and welcome back to week 10 of Applied Immunology. Today we're going to switch topics to talk about immune interactions with tumor cells. As an overall theme in the second half of this course, we've focused on applying what we know about immune responses towards understanding a variety of different types of diseases, including allergy, autoimmunity, and transplantation medicine back in week 9. And we're going to kind of cap off this unit with the last two lectures for this course, which will first examine the interactions between cancer cells and the immune system. Today's lecture 10b will cover the mechanisms used by cancer cells to evade immune responses. We're going to start off today by defining the biological properties of cancer cells, then learn about the important concept of immunosurveillance of tumors, and then end by going over some of the mechanisms of immune evasion employed by cancer cells which promote tumor outgrowth. Before we discuss the interactions between tumors and immune cells, I want to quickly summarize how we define tumor cells. You could spend an entire course just covering an introduction to cancer biology, so this will not be a complete overview, but I still want to make sure that we're on the same page regarding exactly what tumor cells are before we learn more about how they avoid immune responses. If we consider a normal healthy cell here on the left, there are a few steps that must occur in order for it to develop into a cancer cell. Each of these steps requires the acquisition of genomic mutations that are referred to as genomic instabilities. To start off, a normal cell first goes through an initiation phase where it acquires mutations that increase cellular proliferation. The initiation phase then yields a pool of cells that are called precancerous cells, which have proliferated but are considered benign. And the word benign just means that they have abnormal proliferation, but they haven't started to invade neighboring tissue or metastasize to different locations in the body. Some precancerous cells can then enter a promotion phase, where they rapidly acquire additional mutations due to their increased capacity for cellular proliferation. If enough mutations accumulate, these cells can then acquire a new set of biological functions, including the ability to invade other tissues. At this point, they are referred to as cancer cells or malignant cells, which continue to proliferate rapidly and interfere with healthy organ function. So we can think of cancer cell development as a type of evolutionary process where the introduction of certain genomic mutations confers survival or selection advantages upon a subset of tumor cell clones, and this allows them to grow out and form a tumor. This process is often referred to as oncogenic transformation and is characterized by the acquisition of a set of new biological functions that are often referred to as the hallmarks of cancer. These include uncontrolled proliferation and evasion of pathways related to growth suppression, as well as the avoidance or resistance to cell death. This proliferative capacity is also coupled to an ability to replicate indefinitely, so we can think of these as being immortalized cells. Transformed cells also are able to induce angiogenesis, or the synthesis of new blood vessels, and this is critical for establishing their own blood supply to sustain a solid tumor. Importantly, malignant cells have acquired the ability to invade tissues, both those immediately surrounding a primary tumor, as well as through the dissemination of tumor cells throughout the body to form metastatic tumors in new locations. This point is very important in understanding how cancer leads to patient death, since the uncontrolled outgrowth of malignant cells can destroy tissue architecture and start to compromise organ function, and this is what ultimately leads to organ failure. Other biological properties have been more recently added to the hallmarks of cancer. These include dysregulated cellular metabolism, which allows transformed cells to adapt to tissue microenvironments where healthy cells might struggle to survive. Lastly, and most relevantly to this lecture, malignant cells have often acquired the ability to evade or suppress immune responses, such that they can no longer be recognized and killed by immune cells. This process of oncogenic transformation is driven by the progressive accumulation of genomic mutations. So mutations that cause cancer development are often called driver mutations. Not surprisingly, these driver mutations most often affect pathways related to cellular characteristics used to define the hallmarks of cancer. This table from a review article summarizes some of the most commonly mutated genes across a database of the 10 deadliest cancer types. At the top of this list are things like P53, which is a tumor suppressor that negatively regulates cell proliferation pathways, as well as this thing called KRAS, which is a positive regulator of cellular proliferation. It makes sense how mutations in these pathways, as well as those related to cellular adhesion, programmed cell death, DNA replication and repair pathways, as well as immune regulation, could all serve as driver mutations that enable oncogenic transformation. I've listed a few genes in each of these categories that could be considered cancer driver mutations. The underlying cause of mutation in these types of genes can result from two primary factors. The first being inherited mutations in driver genes, which render a patient more genetically susceptible to developing cancer. The other category are external or environmental factors, which introduce genomic mutations. This would include things like exposure to carcinogens through cigarette smoke or radiation, both of which can lead to mutagenesis, or the introduction of mutations into healthy cells. Certain viruses can also trigger tumor development, and we call these oncogenic viruses. These include HPV, that induces cervical cancer, which we've talked about a few times so far in this course, as well as human herpes virus 8, which can cause a cancer in immunocompromised patients called Kaposi's sarcoma. And lastly, hepatitis C virus can cause chronic liver damage and fibrosis that eventually leads to liver carcinoma development. The combination of driver mutations and environmental factors responsible for cancer development can get pretty complicated, and so these things are kind of beyond the scope of this course. But I just wanted to introduce some of these underlying biological aspects of tumor cells before we dive into learning more about their specific interactions with the immune system. So, given this very basic amount of information that we just learned about cancer cells, it seems that at any point in time, a given individual would constantly accumulate genomic mutations that would enable oncogenic transformation, simply as a result of inherited gene mutations, as well as constant exposure to mutagenic environmental factors like sunlight or cigarette smoke or other types of carcinogens. If this is the case, then why are all individuals not extremely susceptible to tumor outgrowth? So to address this, back in the 1950s, two researchers named Frank Burnett and Lewis Thomas set forth this theory called immunosurveillance. This theory proposed that the immune system was able to screen, recognize, and kill off malignant cells at very early stages of growth, before these cells had a chance to form invasive tumors. Based on this idea, one would hypothesize that immunodeficient animals that are missing components of their immune systems should be more susceptible to tumor growth. This hypothesis is fairly straightforward to test using mouse models. One way to form tumors in mice is to inject them with a carcinogen, such as this molecule called methylcolanthine, and it's abbreviated as MCA. If you administer MCA subcutaneously into a mouse on its side or its flank, then in a few months it will develop a flank sarcoma, which is a type of soft tissue tumor. In this experiment, two cohorts of mice were injected with MCA. One were wild-type mice with fully functional immune systems, and one were RAG2 knockout mice. Remember that RAG2 is one of the recombinase enzymes required for VDJ recombination, so a RAG2 deficient knockout mouse is lacking any sort of mature T or B lymphocytes. Because this mouse is essentially missing its adaptive immune system, we can then compare the incidence of MCA-induced sarcoma formation between wild-type versus RAG2 mice, just to see if immunodeficient animals are susceptible to tumor growth. This was exactly the case. So if you look at the percentage of mice with sarcomas along the y-axis and the days following MCA injection on the x-axis, you can see that the RAG2 knockout mice in the black diamonds develop carcinogen-induced tumors at a much higher frequency compared to wild-type control mice in the black squares. There are obviously many other experiments done that have proven the role of individual types of immune cells and signaling pathways in terms of controlling tumor outgrowth, but I think that this study really nicely and simply demonstrates this key concept of immunosurveillance. If this mechanism of immunosurveillance worked perfectly, then animals would never develop cancerous tumors, and we obviously know that this is not the case. If you noticed on the tumor growth figure on the last slide, the immunocompetent wild-type mice still eventually developed MCA-induced sarcomas. These just developed with delayed kinetics compared to the RAG2 knockout mice. So we obviously need to add some layers of detail or nuance to this theory, and we can do this by defining tumor-immune interactions that correspond to a few different stages of tumor growth, And these go beyond the initiation and promotion phases that we first introduced at the beginning of this lecture. This first stage is something that we refer to as the elimination stage, where cells have undergone oncogenic transformation and are then immediately recognized and targeted by immune cells. In this figure, we have numerous immune cells that are capable of responding to the cancer cells that are shown here in red. And these include various T cell subsets as well as NK cells. We can think of this figure as representing an ideal situation, which would be the case if immunosurveillance worked with 100% efficiency and tumors never developed. If 100% of tumor cells are not destroyed in the elimination phase, then we can progress to something called the equilibrium phase of tumor growth. Here, tumor cell proliferation leads to the development of new cells with new genomic mutations that confer a survival advantage to that tumor cell. We can think of these as escape variants of tumor cell clones, which essentially survive the selection pressure that is exerted by the immune system. This can be accomplished by avoiding or actively suppressing immune cells or pathways, and we're going to discuss some of those later on. I think that the equilibrium phase is helpful to think of as a form of tumor cell evolution, where individual clones who are more evolutionarily fit in terms of surviving the selection pressures of the immune system are selected for over time. In this figure panel on the right, this concept is represented by the tumor cell clones in lighter shades of pink, which have acquired genetic mutations that render them resistant to killing by NK cells, something called gamma delta T cells, as well as CD4 T cells. In the equilibrium phase, there are no overt signs of tumor burden and associated disease, which is why we think of the immune system in tumor cells being in this tenuous type of equilibrium. However, there is the development of immune-resistant tumor cell clones, or variants. Once a variant develops that is capable of completely avoiding immune recognition and killing, the tumor can then enter the escape phase of its growth, and this corresponds to the promotion phase of tumor growth that we first introduced. This can be accomplished by either becoming completely resistant to immune cell targeting, or by recruiting and co-opting immunosuppressive cell subsets, such as Tregs, that can basically block tumor cell killing by cytotoxic lymphocytes like CD8s or NK cells. In this phase of promoting tumor growth, the tumor cells are no longer held in check by the immune system, and since they have such high proliferation rates, they can expand and form a tumor mass, at least in the case of solid tumors. At this point, tumor growth is unimpeded, and clinical signs of disease can manifest once the tumor mass starts to invade neighboring tissue to an extent that starts to interfere with organ function. The tumor immune interactions that take place in the equilibrium phase of tumor outgrowth select for tumor clones that can eventually reach the escape phase. This process is therefore referred to as cancer immune editing, or immunoediting. Here, the various cell types and mechanisms that they engage to kill tumor cells essentially shape the biological properties that are selected for in the resulting tumor. The fact that adaptive immunity is involved in tumor surveillance indicates that at least some portion of anti-tumor immunity is mediated through antigen-specific mechanisms. This is a bit of an interesting concept because a tumor cell originated from a healthy self cell, which should not activate the adaptive immune system since immunological tolerance has been established. However, over the course of oncogenic transformation, certain antigens can arise that enable adaptive immune recognition. These can consist of either a new type of molecule expressed by transformed cells or self-antigens whose expression has been dysregulated, and in either case we refer to these broadly as tumor antigens. Because CD8 T cell mediated cytotoxicity is often referred to as one of the main mechanisms for killing tumor cells, the diagrams in these examples will mostly focus on tumor antigens that are processed and presented on MHC class 1. This panel represents the first category of tumor antigen, which are those that are expressed by transformed cells but are not found in healthy cells. These are called neoantigens, or tumor-specific antigens. Neoantigens are peptides that are presented on an MHC that are unique to the transformed tumor cell. One example of this are the novel oncoproteins E6 and E7, which are expressed following human papillomavirus infection. So this means that E6 and E7 are only found in cervical carcinoma cells that have been transformed following infection with this oncogenic virus. So these proteins are not expressed by non-transformed healthy cells. Neoantigens can also consist of normal self-antigens, that have been modified post-translationally through moieties such as glycosylation. This is the case with a modified version of mucin called MUC1, which is actually under-glycosylated in tumor cells compared to healthy tissue. Therefore, this specific MUC1 epitope is only presented on MHC by tumor cells in some types of breast or pancreatic cancer. The other categories of tumor antigens are those resulting from dysregulated expression of non-mutated proteins in a way that differentiates them from healthy cells. Keep in mind that these self-antigens are not true neoantigens since they aren't a novel epitope that's presented strictly by tumor cells. One example of these are melanoma-associated antigens, or MAGE, that's shown in the center panel. MAGE expression in transformed melanoma cells results from the reactivation of certain embryonic genes that are not normally expressed in mature differentiated cells. MAGE falls into a certain category of these called cancer testes antigens, which are encoded by genes that are typically only expressed by male germ cells that are found in the testes. However, due to the overall dysregulation of gene expression in cancer cells, these antigens are now expressed in tumor cells in cancers like melanoma. Another example of dysregulated antigen expression is outlined in the right panel here, and this consists of normal self-epitopes that are massively overexpressed in cancer cells. In this case, the high level of antigen expression means that the density of peptide presentation on MHC class I is increased, which can better support T-cell activation. Examples of tumor antigens resulting from overexpressed self-peptides are HER2, which is found in many types of breast and ovarian carcinomas, as well as Wilms tumor antigen 1, or WT1, which is expressed in some leukemias and certain types of solid tumors. This figure from the textbook summarizes 3 major categories of tumor antigens, which can be divided further into several subtypes. Note that things like tumor-specific mutated proteins, abnormal post-translationally modified proteins, and oncoviral proteins are all considered true neoantigens, in that these are novel protein epitopes. Meanwhile, things like cancer testes antigens, differentiation antigens, or abnormally expressed antigens are all endogenous antigens found in healthy cells, but their expression has been dysregulated in some way specifically within tumor cells. There's a lot of information in this slide, and you don't need to get tied up in memorizing all of these, but I just want to drive home the fact that cancerous cells across many different types of tumors can present different types of antigens on MHC, and that this process is essential for the ability of T cells to recognize transformed cells using their TCR. If tumor antigens are expressed on MHC class I, which makes them available for recognition by T cells, then why are transformed cells not simply targeted and destroyed during the elimination phase of tumor growth? This would represent successful immunosurveillance, but we know by this point that this isn't what's actually observed in humans. The answer to this question is that tumor cells have been selected for based, at least in part, on their ability to avoid anti-tumor immune responses, since mutations that confer immune resistance would give the tumor cell clone a considerable survival advantage. So we can think of each of these processes occurring during the equilibrium phases of tumor outgrowth, and any one of these mechanisms may give rise to a tumor cell variant that's then able to enter the escape phase of tumor growth. There are many different mechanisms used by tumors to avoid the immune system, so let's go over these major categories one by one. Again, because T-cell mediated tumor killing is one of the most important mechanisms for eliminating cancer cells, we will primarily focus on mechanisms that influence T-cell activation and function, but generally keep in mind that additional strategies are used to subvert other immune cells, such as NK cells. First, tumors can be poorly immunogenic, meaning that they do not activate antigen-specific adaptive immunity. An example of this is shown in the left panel, where a tumor has down-regulated expression of MHC class I, as well as other co-stimulatory ligands that promote TCR activation. This is also shown in the microscopy image of a human prostate cancer tissue section here on the right, where you can see that the majority of tumor cells have lost expression of the MHC class I protein, which is stained in brown. Here, the lack of peptide MHC would make it impossible for T cells to recognize the transformed cells. Note that these cells that lack MHC class I would then be susceptible to NK cell killing, but many tumor cells have also acquired mutations that allow them to evade NK cell recognition, in addition to avoiding T cell recognition. A second mechanism of immune evasion would be the tolerogenic polarization of antigen-presenting cells, such as DCs, so that they can no longer activate effector T cell responses. In this situation, tumor antigens would be loaded for cross-presentation on MHC class I, but tolerogenic signals from the tumor microenvironment would result in the APZ presenting antigen on MHC class I, but in the absence of co-stimulatory molecule expression, such as CD80 or CD86. In this situation, the dendritic cell could still travel to the lymph node and prime CD8 T cell responses, but in the absence of co-stimulation, this actually relays a tolerizing signal to the naive T cell, so it doesn't undergo classical inflammatory activation the way that we learned about back in week 4. This means that the T cell would not be able to migrate to the tumor to exert cytotoxic effector mechanisms that would kill tumor cells. The third mechanism is something called antigen escape or antigenic modulation. Here, even if a T cell response has been successfully activated, those T cells can still only kill target cells that express that antigen. Given that a proliferating population of genetically unstable tumor cells should rapidly acquire a bunch of different mutations, the cell population that makes up a tumor can be pretty heterogeneous, and importantly, this includes heterogeneity with respect to antigen expression. Therefore, any tumor cell clone that has either mutated or lost the antigenic epitope used to stimulate T cell responses can now successfully avoid killing by those effector T cells, even if they get into the tumor. The fourth category we will cover today includes a number of active immunosuppression mechanisms used by tumor cells to inhibit cytotoxic immune responses. This includes the secretion of soluble immunosuppressive cytokines, such as TGF-beta and IL-10, which inhibit T cell activation. Tumors can also secrete a soluble enzyme called IDO, which breaks down tryptophan. Tryptophan is an amino acid that is essential for immune cell function, so IDO functions by basically starving T cells of one essential nutrient. Tumors can also express inhibitory ligands on their surface, including PD-L1, which binds to the receptor PD-1 expressed by T cells. PD-1 signaling in T cells is a negative regulator of T cell activation, which can override any other sorts of activating signals. And so this would then render the T cell unresponsive to TCR and co-stimulatory signaling. Tumor cells can also recruit and activate Tregs. These cells are very important in the resolution phase of inflammation following infection, as well as for their roles in maintaining peripheral tolerance that we covered in our week 9 lecture on autoimmunity. But their function is essentially co-opted by tumor cells to shut down infiltrating inflammatory immune cells that might otherwise attack the tumor. Collectively, any combination of these factors work by establishing a highly immunosuppressive tissue niche within the tumor. We call this the tumor microenvironment, and these mechanisms of active immune suppression typically make solid tumor microenvironments a very harsh place for immune cells to survive and function. Lastly, tumor cells can also produce molecules that function by establishing a tough physical barrier that encapsulates the tumor. This includes secretion of collagen, which when deposited at the margins of the tumor can form a barrier that physically excludes immune cells trying to get in to access tumor cells. This is a major issue with solid tumor masses and makes them even more difficult to treat with some of the immunotherapies that we will discuss in the next lecture. Looking at all of these mechanisms together, it's kind of incredible just how many strategies tumors have evolved to subvert anti-tumor immunity. Keep in mind that for tumors that have successfully escaped equilibrium and progressed to the escape phase of their growth, they likely employ several of these mechanisms at once. Considering all of these mechanisms used by tumor cells to avoid immune recognition, why hasn't the immune system simply developed more ways to counteract these strategies? I think that when we consider this question, it's important to keep in mind that, with the exception of pediatric cancers, most cancers develop later on in life, often after humans have reached sexual maturity. Up until recently, the life expectancy for humans wasn't all that long, and until the modern developments of things like antibiotics, vaccines, and other medicines, the pressing threat over the course of human evolution has primarily been infection and not cancer. Given this pressure, the human immune system has mostly evolved to deal with infectious pathogens. So this is reflected by the fact that the mechanisms of killing tumor cells during immunosurveillance are really just another application of the same mechanisms that are used to kill infected cells. From an evolutionary perspective, it kind of makes sense that the immune system can be essentially outrun or tricked by these mechanisms of tumor immune evasion and suppression, because the issue of late onset cancers just hasn't been a threat that needed to be addressed over the course of evolutionary time. This is a bit of a complex overall point, but I think it's helpful to consider when thinking about why cancer is such a difficult disease to treat. So this is just an interesting thing to keep in mind as we head into the remaining lecture for this week on cancer immunotherapies, which attempt to circumvent this issue by boosting tumor-specific immune responses. In summary, today we've established that although transformed cells usually start off expressing tumor antigens that can be recognized through the process of immunosurveillance, cancer cell clones are eventually selected for outgrowth based in part on their ability to evade tumor-specific immune responses. We started off by defining stages of cancer development and the hallmarks of cancer, as well as common genetic mutations that can drive oncogenic transformation. We then introduced this concept of immunosurveillance of tumors and how this leads to immunoediting of tumor cell clones that eventually selects for escape clones. We ended by covering major strategies used by cancer cells to avoid immune recognition and elimination of tumors. These mechanisms will be important to keep in mind for our final lecture this week, where we will learn about cancer immunotherapies that are designed to override several of these strategies of immune avoidance. Today's talk was obviously centered on viewing cancer cells in the context of an immune response, but keep in mind that cancer cell biology also obviously involves numerous immune-independent mechanisms. So, although obviously for this course we approach cancer from the perspective of tumor-immune interactions, this is really only one piece of the puzzle in our understanding of cancer. That's it for today, but please remember to watch the remaining lecture for this week on cancer immunotherapy.